My name's Chris Lloyd. I'm Secretary General of RUMA, and welcome to today's Lungworm, a changing challenge for cattle farmers and vets. And I think the last few days, the news from MSD have just demonstrated how relevant that title is in terms of the changing challenge. What we hope to do this afternoon is to explore the challenge that lungworm is in cattle. We've got experts here this afternoon who've got first-hand experience of that. I'm very pleased that RUMA's been able to host the webinar this afternoon in collaboration with COWS, Moredun, BVA and BCVA. We will address the recent news if you haven't heard it, but MSD, the manufacturers of Bovilis Huskvac, have announced just in the last few days that they're unable to continue production of that particular vaccine, which at the moment we're understanding will cease at the end of the 2027 season. So that will have a significant impact if that situation results at the end of 2027. When we were planning this webinar, that certainly wasn't something we were planning. So we're going to go ahead with the webinar as planned. The history, the lungworm as a worm and how it impacts us. And then we've got some vets here to talk about case studies and the advice and the questions and the thought processes they go through in terms of dealing with lungworm as a problem on cattle farms. As mentioned, cameras and microphones are muted and off for delegates who are joining the webinar this afternoon. That just allows the presentations to flow smoothly. We are recording the webinar, and we will send an email out shortly after today to let you know that the webinar has been published on our website and can be viewed in your own time. So if you're unable to see part of the webinar this afternoon, there will be a chance to do it on catch up, as BBC like to say. Just to repeat, the Q&A function is open, so if you have any questions that you think of during the course of the presentations, please post them in there. When we get to the panel session, I will attempt to go through those questions and ask the panel for their thoughts. We recognise that given the news of the Huskvac withdrawal, advice may develop over the coming months, and so we might well return to this subject again in a future webinar as we come to terms and think through the impact that not having a vaccine might make for the industry. So we have a packed agenda this afternoon. I'm really pleased to be joined this afternoon by Dr. David Bartley from the Moredun, Rob Howe, a vet who's a specialist advisor and has specialised in this area, and Anushka Marsman, also a vet and also somebody with specific practical advice on lungworm. And just at the end of the webinar, I'm just going to hand over to Sarah Gregson, who's the COWS manager, to just give us some sort of insight into the information that is available from COWS, who are the specialist organisation in this area for the industry. We'll go through the presentations and then we'll come to the panel session. So let's get started. I will hand over to Dave Bartley from the Moredun. Tell us about lungworm and why it's such a challenge, Dave.
Right, thank you, Chris, and thanks for the invite to be here. Right, hopefully everyone can see that.
Yeah, that's coming up. Just need presenter mode, Dave.
Yep, that's fine. Excellent. Right, well, thank you very much. Brilliant, good. Yeah, so I've been tasked with sort of an introduction to lungworm. My name's Dave Bartley. I'm a parasitologist at the Moredun Research Institute up in just south of Edinburgh. And yeah, I will tell you a little bit about the parasite. Yeah, Dictyocolus viviparus, that's its Latin name, but it's known as a whole bunch of other things, you know, lungworm, husk, parasitic pneumonia, verminous pneumonia, and parasitic bronchitis all really relate to the same thing. And this picture on the right-hand side is very much what they look like, either in situ in the lungs or under a microscope. Yeah, it does what it says on the tin. It's a roundworm that ultimately infects the lungs of cattle. Different ones cause disease in sheep and deer and other ruminants. It is slightly different from some of the other roundworms that we think of. It can cause significant effects in all ages of animals. It is predominantly one in that sort of first grazing season, but we are also seeing it as a problem within adult cattle. As I say, we often think about roundworms and that sort of direct life cycle, and lungworm is no different, and it has that direct life cycle, i.e. larvae off the grass goes in and they grow, they replicate, and it goes out. But there are lots of differences there. And this schematic, I won't spend ages on it because you'll be able to come back to it. But basically, as with others, we have one of the differences is we have larvae passed out in dung pats. They develop to become an L3. And they have an interesting way of being spread. They're quite lazy larvae. They don't like, they're quite sluggish, they're quite lethargic, and basically they need to be either splattered out by rain or exploded out by these fungi, and I'll show you an image of that in a minute. So like most things, cattle, sheep don't like eating near their pats, so they eat the grass that's got the larvae. The larvae are then taken in and they migrate from the guts through into the lungs. Once they've entered the lungs, they sort of develop, and they do that via the lymphatic system. So they're already moving through the animal. They develop into the lungs, and there they become adults. And the adults, the females, pass out eggs that are embryonated and are passed out. And yeah, it's a long-winded system, but it does work remarkably well. As I promised, those who've seen me talk before, I do love to have imagery for these sort of things because I think it tells 1,000 words. So what we have here is a Pylobolus. That's the fungi. That's the yellow and brown thing. And the little wigglers that are on there are the larvae. And we can see them here too. And we can see the larvae on the pats are there. And yeah, basically, it just shows you that, these things need moisture to both climb up the Pylobolus, but also to stay within the fungi. And yeah, they call, for all of you, the fact you're on this webinar suggests you don't need telling, that can cause huge issues in all ages of cattle. Yeah, the symptoms go from mild to severe. They've got that coughing, the nasal discharges, that respiratory distress, that extended position with the sort of rapid shallow breathing, frequent coughing. And they're all clinical and classic signs of these things. But we can see more mortalities. We can see deaths in animals, in young and old stock. And I know Anushka's going to talk a bit more about that, I think. But, if we think that's bad enough, it's made worse by the fact that we have these sort of secondary bacterial infections, as well as poor development within those animals. And yeah, again, there was provided to me by Ben Strugnall, and really it just shows you what these things look like in situ. So they're white, they're long, they are easy seen in that sort of lungs if they're done. So as well as the sort of traditional clinical signs, we also get reinfection syndrome in this, and this is really a hypersensitivity reaction. It's where the animal responds really rapidly and really immensely to a reinfection. It generally occurs in older animals, and it is in those that have seen larvae before, and it involves really that sudden mass exposure to the animals, and then migrating to the lungs, and it's the hosts fighting off that, so it has a rapid die-off of mature worms, and that causes this sort of hypersensitivity reaction. It can be extremely severe. It causes acute disease and death, and that can occur really, really rapidly. it's often considered to be thought of as an issue where cattle turned on to heavily contaminated pastures. So things like pasture that's had young stock on it before, or co-grazing with heifers that are excreting large numbers of these larvae. So again, yeah, it's just something to be aware of as an additional issue with it. What does it cost? As with sort of traditional roundworms, it is expensive if it's not controlled properly. You know, the sort of estimates, as with all these things, it's considered to be about 140 a pound per adult cow in a milk producing herd. You know, and they can lead up to sort of 4 kilos per cow per day loss in milk. It can lead to, you know, a 22-week delay in reaching slaughter weight. And that's where we see clinical signs, but you know, where we don't see those sort of clinical signs, we can see, you know, large drops in milk production. So, you know, it can have an impact on that sort of growth and productivity, but also, you know, where there are known and recorded issues with the sort of reproduction, so you can have increased seminations required per conception. So, you know, it can take more time to get the animals back into calf. Those calving intervals can be stretched. You know, some of the estimates are about 10 to 52 days, and we get those delays in first service to conception, as well as the sort of condemnation of the waste at slaughterhouses, and that can be significant. So yeah, it is expensive in lots of ways. I was asked to talk about the epidemiology. The larvae that I showed you, the larvae that were wiggling around on those Pilobulus, they like warm and damp conditions, so they are affected by climate, and that helps influences that sort of year to year variability, and I will talk a bit more about that in a second. But they do persist on herbage and pasture like other L3s, maybe not as long, but they are able to persist on there. But they can also be in those carrier animals, as I said before. You know, they can be there in heifers, where they sort of inhibit as an L4, and they can wait there for the conditions to improve. We are seeing concerns about that changing epidemiology, reduced lungworm vaccination, greater reliance on long-acting anthelmintics, reduced cattle numbers, meaning that, you know, immunity is lower, and also there's that anecdotal evidence of anthelmintic efficacy, and I think all of those have an effect on the epidemiology and also the way that we manage these things on farms. So, you know, that how we actually have cattle housed and these things. So I think that epidemiology is changing, and it is something that we'll need to consider going forward. These are patterns of cases. So these are the veterinary investigation ones. So these are basically diagnosable cases submitted to APHA and SRUC. So they are a snapshot. It's not a true epidemiological change, but the graph on the left shows the sort of numbers of cases in 2012, 2018, and 2025. And really what I wanted to show was that actually, you know, the variation goes up and down. So year to year, we have variation, and that probably is driven by climate. But we do see that there's no, you know, we still have that peak in that sort of June, July, August, September time. But we do see that sort of pattern going on. And this is the same for lots of other parasites too. We had a webinar about Homuncus, and we see that same idea where we don't see months very often now where we don't have any cases of these things being diagnosed. The graph on the right hand side really just shows it's a mathematical model. It was kindly supplied by John Graham Brown from Liverpool. It was one of his students, and basically the graph just shows the sort of risk period based on the VIDA data, but used generated mathematically. And yeah, as you can see, it spans the same sort of years that I've talked about, and that sort of corsatinas in, but we are starting to see that pattern extending, so that we're starting to see those risk periods extended for a much larger period. And that is important if we're thinking about how we're going to control these things. The other thing that's from there is that sort of thing of, well, where are we seeing it? Again, some of the work that's done by John and his colleagues and his PhD student would suggest that we're getting a compression of where we're starting to see these things. And these maps, again, using the VIDA data with all of the caveats that I said before. But so these could be single cases or lots of cases. But again, it basically shows that there is a varying pattern seen throughout the years. And that is being now selected. And the thought is that it will become a big issue in Scotland and Northwest, Wales and that southwest corner. So again, it's something to be aware of, but that does not mean that it's not going to be an issue in the other parts of the world as well. And with changing climate, we may see that changing as well. So if we're thinking about immunity, it is biphasic. It's two-phase. The animals need to deal with incoming larvae. So they've got to stop those from establishing, but they also need to get rid of those that have established, because, as I say, if they start to migrate through the lymphatic system to get to the lungs, they can see them at the guts, then they can see them on the way through the lymphatics, and then, as I say, so they have a migratory bit, and then they have that that incoming bit and the established part. And the sort of immunity for these two things differs. So immunity can develop really, really quickly. It is dependent on challenge. So it's challenge dependent. If they see larvae, they will develop immunity quite quickly. So that can be as short as 10, 14 days, but it can take months if it's trickle fed. But the counter from that is it can be lost quickly too. So it's thought that the immunity and some of the work that was done a very, very long time ago, as you can see in the 60s, suggests that the immune memory for those incoming larvae is not very long. It can be a handful of months. So animals need to have that constant exposure to stop that establishment. It's thought that if we look for the immunity for established adults, they actually, the sort of mechanism for that remains there for a longer period of time, for a couple of years. So immunity is not, it's not straightforward, but it is like the other nematodes in that you need exposure in order to develop it to it. And that's the same for all pathogens. Why does immunity not always establish? So as might be guessed, lack of exposure. So unfavorable weather conditions. So if it's dry, it's, if it's the weather like we've got at the second, that's not great for larvae on pasture or larvae anywhere else for that matter. Prolonged housing, if things are housed and not exposed, they will. As was, as sort of suggested earlier, that sort of suppressive anthelmintic treatments, if you stop them from coming in and stop them there, then actually they don't develop immunity to it. And stocking rates, if things are low, a stocking rate is low, then actually we don't see an establishment of that immunity either. Sterile immunity generally doesn't occur. We normally see, about 10 to 20% of the animals continue to carry stuff and shed larvae at pasture. So again, they will shed it and will be part of that exposure for animals coming forward. The sort of take home from the sort of introduction is, it is a significant threat to sustainability of UK cattle industry. We are starting to see that changing epidemiology, be that driven by climatic changes, husbandry practices, resistance, management. There's a whole reason, and the parasites themselves, they're exceedingly plastic. They have a lot of redundant genes in there in which they can use to actually developing where they need to be. We do need to consider sort of anthelmintics and anthelmintic sort of control strategies. And I'm not going to push onto that because I know that Rob and Anushka are talking about these. But if we are thinking about it, and I put these in really around the sort of idea of importing stuff, you know, are we using treatments? Do we need them? And as with roundworms, are we using them correctly? Are we applying them correctly at the right dose rate? Are we storing them properly? You know, and again, we want to make sure whether or not these things are working. So that really just leaves me to say thank you to people who fund me, so that allows me to do these things. And that is me. So thank you very much for your time.
I'll add my thanks to those people that fund you, Dave. That's fantastic information, and we certainly look to the Moredun for their expertise in this area. So, thanks very much for your contribution. I'm going to hand over to Rob to give us a vet's perspective, and just to remind anyone, if you've got questions, please post them in the Q&A. When we get to the panel session after the 3 presentations, we'll come back and review the questions. Dave, Rob, we asked you to come along and give us a vet's perspective. So looking forward to hearing that. Over to you.
Thank you, Chris.
You were sharing. I've just lost. There we go. There we are. There it is.
Yeah, I couldn't quite get the. There we go. Good afternoon, everyone. I'm Rob. I'm a farm vet, founder of LLM Vets in Lancs. But I left practice last year. But in my absence, my fantastic vet and vet tech colleagues and the farmers there are continuing an IPM service, which has been going, I think, 6 years now. And thanks for the invitation to speak today. I have a long-standing interest in bringing lessons from sustainable regenerative farmers alongside the science from lovely, brilliant speakers like Dave, who sits with me at COWS on the steering group, ecologists, and lots of other amazing people. Bringing those into mainstream veterinary practice. And that's a mixture sort of the things that we are increasingly referring to as integrated parasite management, or more simply, trying to stop parasite control being reduced to what do we pour on and when. And lungworm is a great example of why all that matters. It sits with the junction with immunity, and Dave's gone through a hell of a lot on the immunity front, grazing, vaccination, diagnostics, and also a good sense sometimes of that panic feeling you get as a vet when the phone goes off and someone phones up and says everything's coughing. Yeah, so I've got a role today to sort of come at it from a practitioner's perspective. I'm also a BCBA board director, and like I said, been involved with cows since I think 2020. And as Chris alluded to, it's probably an extremely timely moment in the year to have this webinar. So I'll start off by, some of you may have seen an email with a great piece of work by Professor Eric Morgan, and that goes into the sort of strategic framework around lungworm control, including some of the things that Dave has been talking about with immunity and integrated parasite control. So today, I think my job is to probably distill some of the absolute critical components of that. But I'm also going to cover on some of the more simple things. I don't want to teach granny to suck eggs, but there may well be some vet students or vets perhaps in parts of the country that don't see lungworm. So it's important to cover off some of those things as well. So Dave has covered this. So just very quickly, sometimes I think it's important to say lungworm will be present and there will be no clinical signs to see. And actually, that's potentially a thing that we want to imagine and encourage, because that's the sort of trickle effect that nature probably had in its mind when it evolved. You know, that just a very low level exposure gains the immunity without developing the clinical signs. So more and more, it may become our role as vets and SQPs advising farmers is to find that balance. But that very rapidly progresses to the classic soft cough, and then a group cough with the air hunger stance. Some of you, I'm sure, will have seen very severe, and then fatalities are common as well. Dave's mentioned the reinfection syndrome, and from a clinical perspective, it might be worth considering. You might see production losses, so milk drops in that scenario with adults. And a top tip I always find is to sometimes you don't see or hear any coughing if the cattle are still. So just by stepping into a field and they move or walk or run a little bit, gives you an indication of how many are actually coughing and how severely you can instigate the cough. Don't chase them around with a dog, of course. And oh, lovely, one of my favorite videos, this, gruesome. But one of the things I wanted to get across was the difference when we're talking about diagnostics between confirming a diagnosis, and I think that's critical. When we come to treatment, everyone knows if you suspect lungworm, you're going to treat before you are going to have time to get a result, and that's right because of the severity of the disease. But please do make sure you get confirmatory diagnostics done. And I wanted to highlight the difference between that and sort of monitoring diagnostics that we're looking at more and more with integrated parasite management, which is more about, is it in the herd? And perhaps with lungworm in the future, how is immunity doing? So what are our options? We've talked about clinical signs. We've also obviously got post-mortem, and we've seen lots of images already of the lungworms themselves. And as and when we suspect lungworm, the most common test is the test in feces, looking for the larvae. And just to be very clear, there are no eggs in the feces to look for, which is one of the problems. So we're looking for these larvae, and it's very simple to do. You can do it in practice, but often we'll send it off to a lab, partly because we don't do it day to day. The rough costs are up there. And one of the key things to remember is that these things can easily die in transit. They actually like to be in sort of not too cold, not too hot, 12 to 14 degrees, I think. So if we don't put them in the fridge, do send them straight away. And if it's Friday, Saturday, Sunday, which often the emergencies are, you probably don't want to be doing a Behrman's in the post because they'll die in transit. More recently, there is now a PCR available, quite expensive, but if that scenario happened, you could collect the feces and consider sending it for PCR. And then we also have serology. Anushka's going to talk about this a lot more later. And I think this perhaps lends itself most readily to the situation, perhaps in the east of the country, where we don't, or the farmer, we've never seen lungworm on a herd, but perhaps we want to do some tests to make sure that's the case annually in case it comes in. There's lots of different ways it can come in. And as we've just heard, climate and cattle movements can quickly change the location of this disease. And when it hits hard. There's also the possibility to do bronchoalveolar lavages. If the animals are sick, that can be a bit of a stress. So it's not commonly done, I would suggest. And whilst you're up at that end of the cow, we're often doing more and more PCRs on nasopharyngeal swabs, looking to separate out the diagnosis for potentially other pathogens as well. So I thought I'd mention those. And more indirect blood work, you may well get an eosinophilia, but it's very non-specific, so I don't think it's particularly useful. There used to be a bulk milk test in the UK. I think it's something that maybe we should look at trying to get to come back because, you know, we're doing a lot of bulk milk testing now for other diseases, so it'd be a cheap and automatic way to keep our eye on things. It'd be quite interesting as well to potentially look at pooling serology because some of these tests are not very cheap, but that would need validation. And I'll let Anushka talk about the MSP Elisa and her work. So we'll come back to that, I think. So summary of the clinical picture. Lungworms, highly pathogenic, can be fatal, and there's no easy monitoring or predictive test, unlike with gutworm. So in my experience, this makes farmers traditionally, in fact, most of our clients were routinely treating at set times of the year, or perhaps when they heard the first cough. But very much we've been pushing towards more and more vaccination, and that has big benefits. I think I've mentioned this already, so I'd love to see monitoring on farms if they don't think it's there, because we can catch it before it comes in. And HusVac has been a cornerstone of integrated parasite management and protecting our treatments. Yeah, just one other thing to say about that. The the farmers, anecdotally, who have shifted from routine treatments to using the vaccine over the, over whenever that's been, have often seen huge benefits to animal health, and they're definitely sort of fall in love with Husvac even despite its increasing price. And I think the two key reasons that might be, obviously this is speculation, but the first is we now know that resistance is here. It's extremely hard to prove, but we've got reports, and I think Anushka also had reported with the VLA back in 2019. So we know it's here, and if, as is commonly the case, a farmer's been using the same product every year repeatedly for however many years, that is quite a good way to generate immunity, and it might resistance, sorry, to the product within the lungworm, and it might well be that that resistance is at a level that's not obvious, but it is having a negative impact. The other possibility is that the timing of the treatment was not perfect, which is almost certainly going to be the case. It's going to be very lucky to time that treatment to not have pathology before you hit them, or it might miss the lungworm completely and be at the wrong time of year. So I think vaccination compared to just routine treatment at a set time is much preferable. And then coming back to resistance, on the CAL's website, we're really trying to encourage vets and SQPs, if there's any suspicion of resistance to any product for any parasite, to report it. It's very simple and easy to do on the Veterinary Medicine Directorate website. So I've covered a little bit about Husvac already, the principal prevention that we've been reliant upon today. It facilitates massive reductions in anthelmintics, and we're going to hear a bit more about that from Anushka after me. One of the barriers to it, and this is something we'll go through, it is obviously the cost of it versus treatments. There are other situations like spring calving herds. It can be problematic, but there are ways around that. And obviously it's an opportunity for practices or trade to sell it. And I've got an app that Chris Hudson made to help practices look at developing an IPM service and how they might fund that, because it does take time. Another couple of top tips is about the yellow applicator devices, because one of the complaints is the faff of having to deliver it, and also sort of framing it a bit like with some of the sheep vaccines, that it's a lifelong investment if the animal is going to stay on the farm. The second big prevention I wanted to talk about was sort of rotational grazing and pasture risk. And many farmers and vets now are getting a map of the farm and trying to very simply label the risk on each of the fields. And from a gutworm perspective, that might be with fecal egg counts. And from a lungworm perspective, that might well be, we had clinical lungworm in that field in 2026. We know that's a high risk, and we're going to do something different to that next year. We're not going to graze our naive animals, for example. So information is power. And the third thing is all of the life that exists in the dung. And these are a group of dung beetles. Getting rid of a cow pat in about 24 hours, it's actually a mass emergence. But if we have healthy dung, ecology for rapid dung removal, not limited, not damaged by some of the treatments we use, then effectively we get rid of the dung, we get rid of the parasites within the dung, and we have less pressure on the stock. So it's almost a positive circle. And very similarly, developing solid immunity, as Dave mentioned, is also facilitated by using fewer treatments. We have to do this carefully. And I've got a little, the concept of time of effective contact between the host and the parasite is what I'm about to try and talk through, and the potential negative effect of the treatments on the immunity that we're trying to generate. So in this slide, we've got the grazing period marked green, perhaps that's 5 months. And if we use a product that lasts potentially 5 months against a parasite, then we have removed all of the time of completely effective contact between the host and the parasite. Conversely, as an example, if someone used to pour-ons that might last roughly a month. That would represent 40% of that grazing season, or just one, 20%. And if we use one of the other groups where the levamisols and benzimidazoles only last on the day of action, on the day of administration, then that's going to be a much, much lower impact on that time of effective contact. And I'm just picking one example from an IPM planning tool to find out what farmers are doing before you start advising and understand. You've got to understand their system. One aspect is obviously what pasture you turn them out onto. How risky is it, depending on what happened with the stock the previous year? But also the next question is, well, how often are they going to move from there? So if they're set stocked, that's high risk. And if we go from left to right, obviously the more moves that are made, the more avoidance we're building into the system for pretty much all parasites. And I've pulled this diagram and bit of research is actually from talking about gutworm and from Wormwise in New Zealand. But I think we really need to start thinking about this on the left, sort of, rather than we have to make sure that our clients understand that we can't get rid of parasites, they're here. And we obviously want to avoid any clinical effect, but somewhere that subclinical effect and production loss, we're going to be in that region somewhere. And it's a balance between generating immunity and everything else. So I think they're conversations we need to be having more and more. Dave also mentioned this, which is the weather basically, and the lungworm loves warm, wet seasons. And for various reasons, if it suddenly rains after it's been warm and dry, you can get basically a mass emergence of the lungworm, and that can be a serious risk. So I think if we're trying to avoid treatments routinely and we don't have the vaccine, that concept might make the provision of pastures that we can turn stock into that we know are safe at a moment like this would be really useful. I think it's also important to mention always trying to avoid dose and move. So if we do need to treat, perhaps you move first, you allow a couple of days for parasites to fall onto the clean pasture and then you treat. With gutworm, there is a lot of evidence that shows that condensed tannins in things like chicory, plantain, so diverse swards, herbal lays, and hedge and tree browse can help us significantly control the impact of gutworm. There's far less evidence for lungworm, but I guess that lack of evidence doesn't mean it doesn't have an impact. We can't rely on it, but it's something to think about. And compared to a monoculture ryegrass sward, it's likely that either the condensed tannins will help lungworm by holding off the gutworm a bit, and or helping with the lungworm, although to be clear, there's only one paper I could find that talked about it. And you may or may not have seen some of this. This is just a lovely piece of research looking at the content of willow, which is very palatable, and to make the case for both the protein, the mineral, and the condensed tannins content of some of these other plants other than just grass to help with the health of our stock. So a summary, because that was quite a lot there. I've tried to pick out some of the key things. Vaccination, traffic light system, rotational grazing, avoid putting most at risk stock or naive stock onto risky pasture. Be prepared, so try and find ways to have some clean pasture to move stock to. Think about the seasonality and the weather, and I think we're going to need to be getting creative and cognizant of immunity and trying to avoid routine treatments as we go forward. So last section is a little bit on treatment. We're very lucky. The 3 groups that we have of anthelmintics in cattle, they will all work for lungworm, unless there's resistance, obviously. The main thing to say, I suppose, in my experience, and Anushka and I were chatting earlier, everyone is using the pour-ons because they're quick and easy and simple, and no one really wants to have to drench a big cow or a stirk too much. So yeah, the main considerations, I suppose, clinically are if you want persistence of activity, then the MLs are good. So an ivermectin boron treatment is going to be good for that. Always think about not dosing and moving. And if you've used macrocyclic lactones for years and years on a particular farm, or they have, then it might be worth thinking about using one of the other groups for reasons of resistance. The other key thing that I think everyone sort of remembers about lungworm is if there's a lot of lungworm in the lungs and we suddenly kill them, that can be a lot of protein in the lung for the animal to deal with. So it's really important to have a conversation as soon as you diagnose or suspect lungworm and are going to treat. So at the time of diagnosis and prognosis, to have the conversation to say, look, some of these animals are going to essentially get worse when we treat them, don't really have an option. And so obviously the provision of supportive therapies like antibiotics and anti-inflammatories and other methods are useful both to administer at the time, but also get the farmer to monitor and consider adding them in if necessary. And then I wanted to, almost at the end now, just have a think about the different systems. So some of the things we've said about today, comparing different systems we deal with. So this is a suckler herd, suckler calf, and the different experience it has with parasites. And it's much closer to what is in nature than what we do, for example, with dairy calves mostly. So in the interest of time, I'll leave it at that for you to think about, but we can come back to it if we want to. And lastly, just to bring back to what's just happened in the industry, just to let everyone know from a BCVA perspective and cows BVA, we are working to see if we can find a solution to this. No guarantees, obviously, but we all do believe it's a very, very useful product, and it's something that if we can find a solution to keep it going, we should. And at this point, I'll hand over to Anushka. Thank you very much.
Thanks, Rob. I'll get my slides up. I hope you can see this.
I'll give you a shout, Anushka, when it comes up. No.
Yes?
No, not yet.
Sorry, hang on, I'll try again then.
Just while you're pushing buttons, just to remind everybody, what Rob's referring to there was an announcement last week by MSD to say that due to changes in their production facilities capacity and availability, they are not able to manufacture Husvac or guarantee its supply to the market beyond the end of 2027. So it's available until the end of the season of 2027, but beyond that, there's no guarantee of its future. In fact, they're saying they won't be able to provide the vaccine. So that's what Rob's comment was referring to, and I'm sure we'll bring that into the discussion later on. Anushka, that's perfect. That's come up now. I'll hand back to you.
Thank you, Chris. Thank you for the invite. Thank you, Dave and Rob, for your presentations. Welcome, everyone. Thank you for joining. I'm Anushka. I'm an independent vet in South West Wales. I was a founding member of ProStock Vets, but I left last year to pursue my interest in soil biology. The dung beetle project is what I will talk about in this webinar. It was part of my CPD with Vet Sustain, a veterinary approach to sustainable food and farming. And basically, I've had an interest in lungworm in practice for a long time, and I've been involved in this article in 2019, which basically describes a lungworm outbreak in adult dairy cows, and 8 cattle died because of lungworm. It was horrendous. In veterinary terms, the dung beetle project was more an evidence-based approach to challenge anthelmintic usage on dairy farms in South West Wales. So rather than just treat when you think you need to treat, we will try and get some evidence behind it. So the project was made possible by Farm and Connect, First Milk, pro-stock vets and dung beetles for farmers. Hang on, I'll just try. Right. So why did we decide to use dung beetles for this? Well, dung beetles are pretty amazing. They bury and disperse dung. They recycle nutrients and thereby improve soil health. They aerate the dung pads, so they basically make corridors in the dung pad. They dry the dung pad out, and by doing that, they create habitat for other beneficials, and that's what you see, which I come back to later. By doing all this, they reduce pest populations, so you have less flies on the farm and you'll have less worms, gutworms and lungworm. The nice thing about dung beetles is that you can see them with a naked eye, and therefore people can relate to them much easier because a lot of the life in the soil is microscopic and it's not so easy to make it visible. So they're also called a farmer's best friend because they do all these good things. But unfortunately, dung beetles are adversely affected by anthelmintics. So we'll come back to that later. The other thing I wanted to mention is that dung beetles need fresh dung. So slurry is not really an option for them. So if people don't graze, then they won't have dung beetles. And Peter Skidmore wrote this book. It's called Insects of the British Cow Dung Community. And yeah, it's pretty geeky, I will say. But basically it's online, free of charge, you can download it. And what it shows is that the decomposition of fresh dung involves many different insects, and I mean hundreds. Those insects, in turn, are food for birds and other creatures. And I think that is quite an interesting take-home message, that basically fresh dung is an essential part of the wider ecosystem. And if cows or cattle don't go out, we won't have fresh dung. So, and yeah. So back to the dung beetle project then. It was basically a collaboration between First Milk and 31 pro-stock had 40 dairy farms that were supplying First Milk, and 31 of those, so everybody in the invite, 31 farms said, yes, we'd like to take part. So every farm has got a little pot with Farming Connect that they can use. So we use that. And then, of course, the vets are involved as well. Just to give you an idea, the budget was roughly £1,000 per farm. And you can see here the geographical spread of the farms. They're quite, from the Gower down the bottom here, to Tenby, to Cardigan, and up Lamperter way. So how does it work? Well, on the one hand, we have lungworm. We can't really predict lungworm with fecal egg counts because there are no eggs. The female lungworm lays a lot of eggs every day, potentially. But the young stock need to build immunity. But happy days, there is a vaccine. Well, there was a vaccine in 2024. And unfortunately, this week we've had this news. But next year it is still available, and fingers crossed after that. Gutworms are predictable with fecal egg counts to a point. To build up good immunity, we need 8 months of exposure. And in my opinion, once they're immune, there's no need to worm after that for gutworm, but maybe Dave will have different opinion about this. So we did monthly fecal egg counts on 31 farms, and that was quite a challenge. So yeah, it was organization in the practice, but yeah, that was and just get everybody involved. Immunity is really, really important, because if you're not immune, then you will have to fight an infection, and infections are expensive, because white blood cells need glucose to function. And if you think how a ruminant functions, they have to make their own glucose, and milk needs 50 grams of glucose per liter. So any glucose that goes to white blood cells cannot go to milk. And I think that for me is something really important. You need to get your young stock immune before they calve, because if they're not immune, then like these infections are in the population, and yeah, it will cost you milk, because if they're not immune, they'll get infected. And, glucose will be used by the white blood cells. So we decided we'd call it a dung beetle project because it's something different and pique people's interests, So I didn't know anything about dung beetles in 2024. So we brought in Rob. You can see him in the photos on the right here and down the bottom there. So we had a farmers meeting on the 6th of March 2024 to sell the project, because we wanted people to come on board, and also we wanted as many people as possible to use Bovelus Husbach. The following day we had a vet meeting. So at the time there were probably 25 vets in the practice. So all the vets were invited, and we just asked, okay, who wants to take part in this project? And 14 vets said, yes, that sounds like fun. So then the 1st of May 2024, Rob came down and we did a dung beetle safari with 14 vets on an organic farm in the practice, and it was quite good fun. So instead of cows, you can see all these vets looking for dung beetles. So the project was voluntary, so nothing was compulsory. 15 out of 31 farms decided to use Husbach, which was about half of them. We did a questionnaire at the start of the project to find out, what wormers they were using last year and so on. And then at the end as well, the vets then, every vet had 2 or 3 farms. They then went out to do the dung beetle assessments, one in May, June time, and the second one in September, October. Every farmer was allowed to do unlimited monthly fecal egg counts, and it was quite nice because a lot of people were also worming their dairy cows, and I could just say, right, bring in, or they would worm their cows or dry off, and I would just say, well, why don't you bring in some dung and we'll check it. And we never found any eggs in adult cows. We also took a bulk milk sample of every farm, and we looked at antibodies for fluke and lungworm. And within 2 weeks after housing, we took bloods of 10 first grazers, and those bloods were used for antibodies for fluke, lungworm, and we also did pepsinogen. And yeah, that was it. So a bit more about the dung beetle assessment then. Dung beetles for farmers, they have a website, and you can go on there and all the information is there. So they have a dung beetle assessment PAT scorecard, and it's quite difficult to identify dung beetles to species at species level, because there's almost 60 different dung beetles in the UK, but it is doable to identify them by functional group. So you've got Ontophagus, they look a bit like an egg. Then you've got the geotrupits or door beetles. They're very large, they're about 1 inch long, and they're what most people think of when they think of dung beetles. And then there's a large group called the Aphrodinus, and they look a bit more pill-shaped. Now I've got a little video here. The first dung PAT you'll see is this one, and it's taken on a farm that uses long-acting wormers, and you will see there's no dung beetles in this dung pot, and it's not been dried out, and there's lots and lots of fly larvae there, which will become flies over time, which is something that we prefer not to see. And you can see the yellow dung flies. So you can see the fly larvae there. There's loads of them. Well, if the dung beetles were, this is the second dung pot, then with the dung beetle, I didn't get as many as Rob, but you can see a little red one there. And yeah, just to manage expectations, if you're going to do this yourself, you won't find loads. But these dung beetles work on the interface of soil and dung, a lot of them. And the moment you turn the dung over, they will just crawl away. So the vets had these trays, and we would just collect some dung, put it in the tray, add some water, and then because dung beetles are aerobic, they'll float up, and then you can see, you get very quickly a good idea of how many dung beetles you've got and what the diversity is. And then that's not a dung beetle, but a rove beetle. It's part of the insects. And I should have worn gloves, but too late now. So you can see then you get different dung beetles out of that dung pot. Oh. So the fecal egg counts were done by VetPak's second generation. There's different options on the market. We chose this one because it was a sustainability project, sustainability study, zero waste, looking at the literature we felt was most accurate. And last but not least, it gave the best margin in money terms. So we had a system. We managed farmers' expectations. They were not going to get instant results with of their fecal egg counts. So we would prepare the samples as they come in, leave them overnight in the fridge, and run them the following day. And this was all done by the support staff. So I didn't really get involved much. And then the results were emailed to the vet and myself. So in a nutshell then, results. Well, the good news was we found dung beetles on all the farms in the project. The people that used Huspak saw a massive reduction in wormer use, and no one had to worm more than once. No one had to worm their adult cows, but we saw big differences in numbers and diversity of dung beetles on farms. And yeah, if we look back then, after we had everything, after we had all the results, you could see that the low numbers and the diversity were linked to the use of long-acting wormers. And some of the unvaccinated farms had problems with lungworms in the second half of the grazing seasons. And well, we'll come back to this later. So we took 10 blood samples, and 2/3 of the unvaccinated farms had at least 1 in 10 of the first grazers seropositive, so exposure. So this was all farms. On the right, you can see the white part of the graph. This was the number of times that farmers wormed in 2023. So this is first grazers, second grazers, milking cows and dry cows, and the total number was 129. Of the 129, 124 doses were done with mLs and 5 with benzimidazole and levamisole. In 2024, which was a year of the project, the wormers were used a lot less, and the overall reduction on all the farms, Huspak or not, was 65%, which I thought was pretty good. If we look at the Huspak users, yeah, if we compare 2023 with 2024, we saw an 86% reduction of wormer use, which is amazing, I think. And yeah, it just goes to show that we can challenge it. But this is also thanks to the HUSVAC. So it can still be done next year, but after that, it's questionable. So does it stack up financially? Well, the biggest costs are in red. So if you scenario 1 is no vaccine, if you do get lungworm, I could only find one study that's quoted 50 to 100 pound per animal. This study was quite old, so you could ask yourself with the price of cattle and that this is a bit more than this. On the scenario 2 where we use HUSVAC, the HUSVAC is the most expensive part, and yeah, in 2024 it was about £9 for 2 doses. Well, in 2026 I bought some myself. It was about £11 a dose. But still, if you do get lung damage, it's the scenario 2 still, it's an investment, but yeah, it's worth it, I think. So results for lungworm then. The bulk milk antibodies were negative on all farms. So none of the farms had a positive bulk milk. But this doesn't mean that individual cows are negative. So there could still be some cows that are positive. The first, we blood sampled 10 first grazers, and we checked, the ELISA we did was the major sperm protein ELISA. And what that means is that HUSVAC, the larvae in the HUSVAC, they have been damaged, so they'll never become adults. They never become fertile. So the vaccine lungworm will not seroconvert major sperm protein ELISA. And the wild type lungworm are fertile, and they will trigger a positive ELISA. Yeah, so we could basically distinguish between those two, which was quite nice. And we call, exposure was if at least one of the 10 was positive on the MSB ELISA. In the vaccinated groups, this was 33%, 1 in 3. The non-vaccinating groups, it was 67%. So of the non-vaccinating herds, 2/3 had, 2/3 of the young stock have seen lungworm. And yeah, there's lots of questions here, how much exposure have they had? Are they immune? And those are questions that will remain. But yeah, I thought it was quite interesting. Like, you could argue the vaccinated group, maybe they were protected enough, so the wild type lungworm never made it to adults. And those are questions I don't know, but there was definitely a difference there. Last but not least, we tallied up all the dung beetle scores, and it was quite nice to see, which I totally didn't expect, is that the people that historically wormed the least, so this is again in white 2023, and then 2024, very little worming, those people had the highest dung beetle scores. And yeah, and if we think that dung beetles only have 1 or 2 life cycles per year, it will take a couple of years to get good scores. I would like to acknowledge a few people and organizations. First Milk, Farming Connect, all the farmers in the project, the support staff and the vets at ProStock, Rob Howe, John Graham Brown and his team at Liverpool University, because they did all the bulk milks and the bloods, and Dung Beetles for Farmers. Joachim Bauer, thank you very much.
But thanks, Anushka, and given that it's Royal Welsh Week, diolch yn fawr to you too. If I can bring Dave back on screen and Rob for the panel session, just to remind you, if you've got some questions, please post them in the chat. I've recorded 2 or 3 that have come in so far. We'll kick off with the questions in the chat, but you've stimulated my thoughts, everyone, as well. So I've got a few if I get chairman's privilege at some point. I think basically we have to acknowledge that the news from MSD last week is going to present some challenges to the industry. And just to confirm again, the news from MSD is that due to changes in their production facilities, they're not able to guarantee the manufacture of Huskvac beyond the 2027 season. So that's the situation as we understand it. Rob, you might want to speak a bit more about this, but I do know that you've been actively involved already with BCDA and BVA and corralling industry support to raise awareness to the issue and potentially look at how we resolve that issue going forward. And I think this afternoon's webinar has just shown how important the lungworm vaccine is, as your presentation just showed, Anushka. So I think I'm just acknowledging that from the questions that are in there. Just to come back to a more specific question in regards diagnostics, are there any new diagnostics on horizon, maybe more affordable serology? This is a multi-part question, so bear with me. Are there any more diagnostics on the horizon? And then there was a specific question to you, Rob, I think, about the fecal PCR lab test that you mentioned. Who's doing? Are we able to say who's doing it? And were they able to look at pooled samples?
I think it's fine. The labs that I've phoned round, I believe the PCR is Axiom, and I think the answer is yes, they can do it across 5 samples. I think it's accredited for 5. If anyone wants to correct me on that, Dave, Anushka, let me know. And then in terms of the other question about future tests, I'd be looking to the serology, and then it's one way to get the cost down is to get it validated for pooled on bloods, and probably the best option would be to be able to test it on milk and bulk milk and potentially individual milk if it was that cheap and it became that big a problem. That would be my initial thoughts, but we're not there yet.
So I was thinking about bulk tests, but Anushka, your project showed that the bulk tank tests didn't indicate potentially cows within the herd that were carrying it. So does that leave us still vulnerable?
Well, the thing is, with these bulk milk analyzers, there's a cutoff point. So it's not necessarily a negative, it's not a 0, but there's just a cutoff point. And it's, it doesn't mean that there's no carriers in the herd, but it just means that, it's not a major issue. But I just wanted to, make sure that everybody understands that if your bulk milk is negative, it doesn't mean that if you have 300 cows on your farm, there could well be 1 or 2 that are positive. It's just dilution. But there is a cutoff point with these bulk milks, so the result is not 0.
And Dave, given Moredun look into a lot of these things and are involved on the cutting edge of the research, any positive news you could give us
about the diagnostics? There is certainly an interest in doing it. There certainly was a funding application put in, but it unfortunately was unsuccessful. So yes, there are people looking at it. Liverpool are, and others. So yes, hopefully. I think, I agree with Rob. It's likely to be serological, or those pen-side lateral flow types of devices that work if that's the case. So it's worked with Fluke, so there's no reason why it wouldn't necessarily.
So that would give us an indication of presence, but just moving to the immunity side of it, is there any? I've learned this afternoon that building up immunity in the herd and particularly in the young stock is really important, and we'll come back to that in a minute from a strategic point of view. But is there any way that we could measure that to get an idea of what immunity has been built up? But how long would it last? You're telling me it could be quite short-lived, so maybe the test isn't valid. or useful to us. So my question is there a test for immunity?
I mean, the ELISAs are looking at antibody levels, so that is what you're looking at. But yeah, you hit the nail on the head. It's there's exposure and then there's immunity, and that's the thing from it. So yeah, and as in my slides, that immunity for incoming larvae is actually quite short, or can be if they're not exposed to it. So yes, I think it would be. I think it's going to be a thing of a bit like Rob's talking about. That monitoring, risk management, that sort of thing of understanding what's gone on, what the clinical signs are. I would sense.
Up to date, my biggest thoughts and use about the monitoring side for lungworm is for areas of the country where they think they don't have it, and just to be sure of that, I think we've basically either been relying on treatments or vaccine where lungworm's been a threat. So I don't yet have the experience to know or to have a gut sense of what the tests may or may not be able to tell us about immunity, because it's very complicated, and lots of complications to be sure that there's adequate immunity. But yeah, maybe we'll get there. Maybe we can have another webinar in 18 months time and see where we're at. Well, maybe sooner.
So just to ask the numpty question, so what I've learned from you guys this afternoon, all of you, is that immunity can be, building up immunity is important. The immunity in the young stock perhaps has less stability, so it's could be, I think Dave's quote was, it can be quick to be built up, but can be quick to be lost. So if I'm grazing heifers the following year or beef cattle in a second grazing season, do I have to assume that I start my immunity levels at 0 again and I've got to build them up in that second grazing season? Would I be right to assume that, or is there a carryover of any immunity to the second year? Dave, Rob, Anushka, anyone can take.
No. So I thought Rob was going to think. I mean, as with all these things, it very much depends on what they've been exposed to. So if they've been housed and they're not seeing anything, then immunity will start to wane. If they're out and they're being exposed to these things, then that immunity is very much challenge driven. So they have to see it in order to keep it. So yes, if they saw it later into the season, they might carry some of that immunity back over, if you've got 4 or 5 months, but it certainly would be waning. And as with all these things, they would need to re-see it in order to build that back up again. And that is the thing, is it's enough exposure. I think Anushka said it, enough exposure to allow them to develop immunity, but not so much that we start to see clinical signs.
I think turnout, like in 2024, it was very wet. People didn't turn out until June. But then, on the other hand, we were still sampling young stock after housing at Christmas time. So every year is different. But I think at turnout, it's just important to realize that which pastures are at risk, which comes back to Rob's presentation, and which pastures are not. And I think if you can turn your first grazers out to a low risk pasture, that's a good starting point, because then they can get a little bit of challenge and build up immunity, and then they get a bit more challenge again, and then a bit more immunity, and then all of a sudden it's not a problem anymore. But I think it's just getting farmers to think, and education and all that, and have discussions. Like we had a calendar, but I didn't bring it up because we're only at 15 minutes. But, just to visualize, what's happening, and what are the biggest risks, and reseed, side-dish aftermath, obviously that's the lowest risk. But it's just having that conversation.
And it might be worth thinking, if you think about it from nature's perspective, cattle would be out all year round. And so they've constantly got a trickle dose, and the mothers are with the calves. So the mothers are acting as hoovers, and the mothers are immune. So they're killing off a lot of, or, slowing down the breeding, the eggs. Whereas if we keep calves all together, they're little mini factories of parasites. And if we keep them on the same pasture, they're going to go crazy, which is why we use products to treat them. But equally, I would argue against that because of the risk to resistance of doing that. Same pastures every year, same products, same most at-risk animals. And I think we just have to try and think about what might be going. We can't tell what pasture levels are. We hope, probably the best thing would be that there's a low level everywhere, really low level. It's trickle, trickle, trickle. And the vaccine has enabled us to dampen down the overall level across a farm, the grazing platform, by immunizing the most at-risk animals and the ones most likely to populate the pasture with risky infective larvae. And that's, you can get quite lazy because that does control it brilliantly in my experience. If you read the data sheet, if you've got high challenge, you might need to give them a booster dose in the second season as second season grazers. Touch wood, I don't think I've ever had to do that. I don't know if, no. So
I
don't know if you guys can see the questions, but don't want to get us bogged down on the serology and immunity. But there's a question in there about serology testing. We'll be looking more at the immunity to adults versus short-lived larvae immunity. And then it goes on to say, could we see using serology testing as a signal to treatment? To see if treatment is necessary or to monitor immunity levels. I think maybe we've covered that. Is there anything you want to add to that?
Well, we did in the dung beetle project, we did pepsinogen as well. And we did actually advise people to worm if they have positive pepsinogens soon after housing. But with the lungworm, we didn't really advise because there was no clinical signs. So okay. It was,
I don't want to get us distracted, but I just want to acknowledge there was a question about immunity to gutworms. But Dave, I'll put you on notice. Rob, I'll put you on notice. We may cover that as we might look at gutworms in a future webinar. Just one question, again, slightly tangible, but flukicides, do they affect dung beetles? To the dung beetle specialists on the call? Flukicides, do they have the same negative impact on dung beetles?
Yeah, there's quite a lot of work now that shows they do have a significant impact on dung invertebrates in general, and the impact that then has on the bird life that feeds off those. And the chuff was the classic. Was it Philip Scoose's project, Dave, that looked at that? Yeah. And others. Not that traditionally years ago, the flukicides were grouped within the products that were sort of dung beetle safe. But I think no matter what the product is that we use, it all has an unintended negative consequence in the environment somewhere. So I try to point people away from worrying about specifics. We
could probably have a webinar on dung beetles just themselves, couldn't we, Rob, knowing your expertise? Dave, again, this is probably a stupid question, but when I was looking at that flow diagram you showed us, the life cycle, they are pesky little blighters, aren't they? The way they manage to get from the gut into the lymphatic system and then into the lungs. But if I understood your diagram correctly, the adults shed the larvae while they're in the kind of esophagus, the throat, the tracheal area. Yeah. So does that mean they're passing them onto the pasture through the throat, or is it like we've experienced with gutworms through the feces? So
they basically, the adult worms the females lay the eggs and they develop, and then they're coughed up and then they're passed through. So they go up and then they go through the gut system.
Okay. They do go through the gut, so it's not.
They go back through the gut and then the thing. So they'll. It's one of those things. If you look at it, some suggest that the eggs hatch where they are in the lungs, and others suggest that they hatch as they go through the system. But anyway, the general point is they go back and out into the poop. That's how we end up with the larvae in the poo.
So I think at that point, I think we've kind of gone through the questions that have been presented from the delegates on the call. I'm going to thank you guys for the assistance of the panel. If another question comes in the next few minutes, I might bring you in, but I'm going to just hand over to Sarah, who is the COWS manager, just to sort of share with us the information that is available from COWS, and maybe what I would say is, if you've got any other ideas for topics for future webinars, if you want to place those in the Q&A, we'll happily give those consideration for future events. Sarah, I'm going to hand over to you.
Yeah. Just trying to share, but not failing to.
No, that's okay. You are sharing. Just if you can put it onto presentation mode. That's it. Give it a moment, it'll come through. If not, that's it. Perfect. You're good to go.
Okay, great. Well, thanks, Chris, and thank you, RUMA, for facilitating this webinar on lungworms. I'm not going to keep you long because I know it's the end of probably a long day, long Monday. So I'm just going to run through very quickly for those that don't know about COWS or those that aren't sure what COWS does. We're a set of voluntary independent experts providing the latest evidence-based information, advice on tackling roundworms, lungworm, liver and rumen fluke, lice, mites, ticks and flies. We're caring for, obviously, trying to prevent disease, parasitic disease in our cattle, but also caring for the welfare as well as for the environment and the lovely dung beetles. We're run by a steering group with 30 or so members, and this is a group of vets and academics. We've got a farmer, an SQP, and also organisations, VMD, NOAA, AMTRA, ARDA, HDB, RUMA, NFU, and there might be some more that I haven't remembered. And we meet 4 times a year, 3 online, 1 face-to-face in November. And we were founded in 2010 based on the model that SCOPs that have been very successful in the previous maybe 10 or more years. So our target audiences are vets, SQPs, RAMAs, dairy and beef farmers, the whole gamut. So quite a mix of different messages, well, the same message, but presented in different ways to the 3 groups of people. And we are trying to provide the best available evidence-based information to the beef and cattle industries in relation to the sustainable control of both internal and external parasites. At the core is the COWS technical manual, 5 chapters of the latest information and advice. This is updated every 3 years. The next update is autumn 26, so we've actually started doing this with John Graham Brown from Liverpool University doing the updates. We're also including new chapters on crypto and coccidiosis, which hopefully I think Dave Moredun is going to be authoring. And we've also got a chapter on IPM, which Rob is going to be doing for us. These, the COWS website houses all the chapters, plus internal resources and signposts to external resources. It also has rolling news stories and things to do now on every page. So with content changing at key times of the year, so at turnout, summertime and at housing. We also do social media on X, 2 posts a week. We've got 2,500 followers and we get high engagement rates. And we sometimes go to a few events like the ARDA conference. We do press articles in the vet press, the SQP press, which is actually over the counter. We always have an article in there in all their issues. And we also do the CPD quiz. A couple of years ago, we did a few of those where we put our information in, and then the SQPs did the quiz and then they got CPD points from that. And of course, the farming press, we're very lucky. We've got a lot of publications that will carry cattle stories. And we do regular discussion with members of the steering group and like-minded organisations such as SCOPs. We issue joint fluke forecasts, warnings with SCOPs. And last week we did a whole workshop with SCOPs on liver fluke. So there's lots of collaboration going on across the species. And we're talking about things like testing before treating, halting resistance, and responsible prescribing and admin. We're going to continue promoting the best way to administer foaming products. I think it was mentioned some point that, you know, actually underdosing is a real problem, and so trying to get farmers to dose correctly. We're building a body of evidence to show what resistance there is to cattle worming problems. We know we're not as bad as sheep, but we don't want to get that as bad as sheep, as it were. And we're going to continue to highlight the benefits of IPM. We use innovative communication via webinars such as the one that you've been listening to today, podcasts. And we extend collaboration with like-minded organisations such as BVA, BCVA, RUMA and RH&W. And we're a very small organisation. Our budget is very small, but it's with thanks to these 8 AgResearch, AMTRA, NOAA, ARDA, HCC, QMS, HDB and NFU. Thanks to them for funding for what we do. And I think we punch above our weight. So I don't think we're, if I say so myself, doing too bad a job. And I'm just finally just put in a promo for Rob's course. He's set up this new course on integrated parasite management. This is very much supported by cows. The next one is on September 22nd and 23rd. And if on the recordings or whatever you want to scan the QR code, that will take you through to the form to fill in to attend. So that's it.
Oh, I was just going to say, perhaps you could leave that screen up so if people wanted to look at that. And
okay,
just while I close out, Sarah, thank you very much for that. And obviously, if you want any information on cows, I find if you Google cows parasite, it takes you right to the site. So that's my little tip for the afternoon. Just for me to say thank you very much to Sarah for that little quick update on cows, to Dave, to Rob and Anushka for sharing your experience and expertise this afternoon. Clearly we have a challenge and I think the landscape's going to be changing. As you mentioned, Rob, we may be looking at this again in the future as we've learned to whatever the news, to adapt to whatever the news turns out to be in the long term regarding the availability of Huskvax. So thanks to our speakers this afternoon. Thank you to all of the delegates who've joined us this afternoon. I hope you found it helpful and useful. There will be a recording available of this webinar. So if you want to share it with others or let people know, or if you've missed it, you can get it on catch up. We'll share that with everyone who registered by email later this week. And there will also be links on the cows website, the AMTRA website, where you can also get CPD points, and BCVA sites. So there'll be plenty of links to the webinar. Thank you very much for listening this afternoon. We look forward to seeing you at a future webinar. Thank you very much. Thanks, everyone.